All right, so today we're going to talk about the auditory system. The first thing that we should talk about is that with audition, you have to remember that the auditory senses depend on our ability to detect pressure changes. Now, these can be periodic compressions of air, water, or any other conductive media, even the skull. These pressure waves are then transduced into action potentials. This is sometimes what's referred to as the neural coding, just like wavelengths of light are transduced into action potentials at the retina. Periodic compressions of air usually are transduced into action potentials in the inner ear. The nature of sound is that it has two qualities that we'll talk about. First of all, these changes in air pressure have a property known as amplitude. The amplitude refers to how large the wavelength is, or how large the compressions are. And then the frequency refers to how many of these compressions there are per second. The amplitude is something that we interpret as loudness. So while amplitude is a physical property of the compressions, loudness is a psychological property of these wavelengths. The frequency is a property that relates to the pitch that we hear. So while we'll talk about frequency as being a physical property of these compressions, the pitch is something we refer to as the psychological property, so our perception of frequency. You can see down here in the image just an illustration of the difference between something that is low frequency or has a low number of cycles per second and something of higher frequency with more oscillations or more periodic compressions per second. Amplitude is also illustrated here. So here's a low amplitude waveform or a series of low amplitude compressions and a series of high amplitude compressions down at the bottom. It's important to note, because we'll talk about some of the methods by which the ear codes hearing for humans, that our hearing range is somewhere between roughly 20 to 20,000 hertz. So that would be 20 compressions per second up to about 20,000 compressions per second. So let's take a look at the structure of the ear. There are three main compartments that we'll talk about. The first is the outer ear. So usually when we talk about the ear, this is what we're talking about, is the pinna, or the structure that sits outside the head, and the external auditory canal. So the pressure waves would impact the pinna. We'll see that they are changed subtly by the pinna and the shape of the pinna. And then those compressions of air will move through the auditory canal and impact the eardrum or tympanic membrane, which is the beginning of the middle ear. So the outer ear serves the function of being what's called a frequency filter. And I'll show you in a minute what that means. But it's thought that one of the functions of the pinna is to help with sound localization. By filtering certain frequencies, we're able to make distinctions between the left and right ear, much like we do with our eyes to determine distance and localize in 3D space. We use differences between the two ears to infer the positions or the localization of incoming sounds. So the middle ear begins with the tympanic membrane or eardrum, and then goes to the three small bones of the middle ear, the malleus, incus, and stapes. The small bones serve to amplify the vibrations at the eardrum. So incoming compressions will vibrate the tympanic membrane at the same frequency at which the compressions are occurring. Those compressions then will move these three small bones that will amplify the signal as it then causes compressions of the oval window. It's at this point that the middle ear converts these airborne vibrations to liquid-borne vibrations, because the oval window is a fluid-filled structure. So movement of the oval window will cause these vibrations to be carried through the fluid that's internal to the inner ear or cochlea. So the cochlea then takes these vibrations of its fluid, so there's a fluid-filled canal here in the cochlea, and those vibrations will impact specialized structures in the cochlea, where those vibrations are then converted into neural impulses or transduced. So the important thing to remember here is that the transduction of compressions, that is the properties of frequency and amplitude, those things are converted into neural impulses at the cochlea, which is connected directly to the auditory nerve, which will carry those signals back to the brain. So I need to talk for a minute before I describe in a little more detail the function of the pinna about the idea of the spectrogram and interpretation of frequency and amplitude. So this isn't a great example, but I think it should make the point clear. On the left here we have a couple of different signals, and you can see that those signals vary in amplitude, so some of the waves are big, and they also vary in frequency, so there's some high frequency components to it, and very little in terms of low frequency. So you might have expected a low frequency to look something like this, having very slow oscillation. The representation of this waveform on the right is called a spectrogram, where we have frequency on the y-axis, time on the x-axis, and then amplitude is reflected in the color of the image. This is sometimes called a heat map. So you can see here the bright red spikes correspond with these spikes in amplitude at higher frequencies. The absence of the low frequencies is reflected in the blue region down here, so there's very little in terms of low frequency activity. The reason this is important is you can imagine any incoming sound, which would have a representation like this on the left, can be taken apart into its subsequent frequencies and amplitudes over time, and this is what might be interpreted by the ear. So the reason, again, this spectrogram is important is to interpret this image from your book, where you have frequency again represented on the y-axis, time represented on the x-axis, and then the amplitude represented as color. So what's shown here is an initial sound pattern, and this would be a sound pattern that included frequencies from, in this case, 0 to 20 hertz over the course of 5 seconds with a constant amplitude. So all of this red area, because you don't see any differences there, means that this is all the same amplitude of each frequency. We can then pass this initial sound pattern through molds of an individual's pinna here and record what comes out the other side, or what would have come out into the ear canal. And when this has been done in research, what they find is that the pattern that emerges through the pinna is slightly different from the input pattern, and that it's slightly different when you compare what happens with the two ears. So you can see here on the left ear we have a filtering or a modulation of the input pattern, tending to, in this case, accent or accentuate lower frequencies compared to higher frequencies. And we see the same kind of pattern from the left ear, but it's slightly different. So as these two signals then, after they've passed through the pinna, move on to auditory cortex, it's thought that the brain has been able to, or has learned through experience, to understand these differences, and by virtue of the way that the pattern ends up impacting the brain, can determine from these differences in the amplitude and frequency profiles where in space this sound may have been coming from. It's also the case that I mentioned here that these might be molds of an individual's pinna. There have also been studies where an individual would wear the molds from a second person's pinna. So if you take an individual and put them in a room without these molds, and you ask them to localize sounds, so they may place speakers around the room, people are pretty good at localizing where sounds are coming from. If you, however, take another individual's pinna, or a mold of that pinna, and place it over the first participant's ears, their ability to localize sound decreases. And that's again thought to be because our brains have learned how the output from our own pinna should look, and when we alter that sound profile, we lose some of our ability to locate sounds in space. Next we get to the middle ear, and again I've already mentioned the primary bones of the middle ear. So in the ear canal, these compressions of air vibrate the eardrum at whatever frequency is impacting it. Those vibrations are then transmitted through the three bones, the malleus, incus, and stapes, and those vibrations then cause movement of the oval window. The primary function of this middle ear is thought to be amplification. So not only does it transmit this signal over a very small distance, but it also serves to amplify the frequencies that are impacting the eardrum. Once we get to the inner ear, what we find is that the tympanic membrane, which has now caused these vibrations in the middle ear, vibrating then again this oval window, that this is where the fluid-filled tunnel of the cochlea causes stimulation of tiny little hair cells. So the fluid vibrates in the oval window, and those fluid-borne vibrations then move down the cochlea, and as they do so, they tend to displace tiny hair cells. And it's the displacement of the hair cells, or the movement of the hair cells, that causes an excitement or stimulation of the auditory nerve that is connected to those receptors. You can see here in a cross-section of the cochlea that there are several chambers here, but the important chamber that contains these audio receptors is the basilar membrane. So in the center of this, we find the basilar membrane, which holds the dendrites from the auditory nerves, sometimes here called the cochlear neuron, and those auditory receptors with their little hair cells at the end. Here's an electron micrograph of the inner ear and the basilar membrane. So you can see down here the basilar membrane containing the auditory nerve, and then those little receptors with their hair-like extensions waiting to detect the movement of fluid. It's important to note that about 99% of people who have some kind of hearing impairment do have a response to even loud noises. So even people who might describe themselves as legally deaf probably have some response to very loud noises, meaning a neural response. This distinction is important because it is most often the case that deafness is caused by some kind of a conductive or middle ear deafness. That is a problem of the transmission of the compressions of air from the tympanic membrane to the oval window. Nerve deafness is what's caused when you have damage to the cochlea, and this is a problem of actually transducing those compressions into neural impulses. This brings up the cochlear implant, which is one of the only solutions we have to true nerve deafness. So it's the case with middle ear deafness or damage to the tympanic membrane that we might be able to restore some function of the transmission of those vibrations to the cochlea, provided that the cochlea is still capable of signaling back to the brain. But in the case of nerve deafness, where you have damage or dysfunction of those hair-like cells, the cochlear implant might be required. And the way the cochlear implant works is that it actually sits right along that basilar membrane and contains a series of little microelectrodes. So it can actually use little electric impulses to stimulate those auditory receptors. This cochlear implant is wired directly to a little microphone and microprocessor. And the microphone on the outside of the head picks up noises, just like any other microphone, and then a small microprocessor determines, based on that input frequency and amplitude, how exactly to deliver stimulation to the cochlea. Now, the reason that we're able to do things like this with the cochlear implant is that it's understood that the cochlea conveys information about frequency in part by which part of it becomes stimulated. So there's a theory about audio perception known as place theory, and that is based on the idea that each area along the basilar membrane is tuned to a specific frequency. So when there is input, so vibrations coming from the oval window, those vibrations, when they impact the cochlea, it is the case that very high frequency vibrations will tend to stimulate hair cells along this base of the cochlea, or at the beginning of the cochlea. As it coils around, the hair cells become responsive to lower and lower frequencies. What this means is that you can almost think of the cochlea, the basilar membrane of the cochlea, like the keys of a piano. If you took your implanted cochlear implant and were to stimulate here, for example, the person would tend to perceive a lower frequency. And if you stimulated the cochlea up here, you would tend to perceive a higher frequency. And this is the way that cochlear implant, by using the microprocessor to plan out or program a pattern of stimulation along the cochlea, that it's able to replace the function of those hair cells and restore, in some cases, or I should say in many cases, somewhat of a normal auditory environment for an individual with nerve deafness. Now, one thing I'll mention is that when you think about this, I hope it occurs to you that when we're talking about being able to perceive things between somewhere in the neighborhood of maybe 20 and 20,000 hertz, that there are essentially an infinite number of potential frequencies between 20 and 20,000. And we couldn't possibly have a neuron for every single frequency that we can perceive. So this becomes a problem for the place theory, in that it doesn't really seem possible that we could have an individual neuron for every frequency. Like if I were to stimulate this one little neuron here, I would get exactly 5,000 hertz. So we have to be thinking about other frequencies, or other theories that have been developed to understand how it is that our brain codes these periodic compressions of air into neural impulses. The frequency theory is another theory that is thought to also be functioning at the same time as place theory in our nervous systems. The frequency theory suggests that the auditory nerve produces action potentials at the same frequency as the input compressions. That is, that as compressions impact the cells of the cochlea, that they essentially fire on every cycle of the compression. So if there is a 20 hertz or 20 compressions per second input, that we'll see 20 action potentials per second at the nerve ending. I hope you recognize pretty quickly, though, that there are problems with this theory also. So if you had a neuron that wanted to fire at the same input as it was being stimulated, that might work at 20 hertz or maybe even 200 hertz. But when we get up to the range of 20,000 hertz or 20,000 times per second, neurons simply aren't capable biologically of producing that many action potentials per second. So it can't be the case that frequency theory would account for all of the ways in which the auditory nerve communicates this information about frequency and amplitude to the nervous system. In order to account for some of the higher frequencies, it's thought maybe that something called a volley principle might become involved. So the volley principle is one that suggests that at low frequencies, here we have the input compressions at a lower frequency, that we might have neurons that generate action potentials at the peak of each, or at some point in the cycle of each of these compressions. If that were the case, then this neuron could communicate information about this particular frequency. For higher frequencies, where the neuron isn't capable of producing that many action potentials, it's thought that we might be listening to a collection of neurons. So there might be, in this case, 7 or 8 individual neurons, all of which are firing somewhat in response to this input, but they can't possibly keep up with that oscillation. However, when you add up, or if you have a neuron onto which these things converge, so in other words, it's listening to the output from all of these individual neurons, that collectively their signals might be able to approximate the input frequency. So it's thought that through some combination of place theory, frequency theory, and this volley principle, that we are able to decode or understand these differences in the frequency and amplitude of these compressions of air. Now, one thing I'll mention here is that your book talks about labeled line and across fiber coding, and we'll talk in a minute too about how this compares across the different senses. But if you remember the idea of in vision of having only 3 kinds of color receptors, but using that information to decode a nearly infinite range of colors that we can see, then that would be something called a cross-fiber coding, meaning that we take the the input from a small number of neurons, and we compare across those inputs, and from that comparison of the relative activations of those neurons, determine some higher or more complex kinds of information. So the volley principle is one that illustrates this across fiber coding strategy. That is, we take information across fibers, combine that information, and we're able to decode this higher frequency waveform. The labeled line coding approach to neural transduction is one like the frequency theory that suggests that we would have an individual neuron that communicates a specific piece of information. So a neuron that fires for green, and a neuron that fires for red, and a neuron for every color in between would be a labeled line theory of vision. But of course, we know that that's not the case. In the case of the auditory processing, it's thought that there's some combination of labeled line and across fiber. That is, maybe some neurons do respond according to the frequency theory and communicate a specific piece of information, whereas in other cases, groups of neurons may combine their information across fibers. So what happens after the cochlea? So we have this transduction of compressions of air into neural impulses. Those impulses then go to the ipsilateral hemisphere to what's called the cochlear nucleus. So this is a small nucleus that sits in the brainstem. I hope you can see that this is just about the level of the pons. So this would be the posterior end of the cerebellum that's been cut away in this case. And we can see here this dorsal part of the hindbrain sitting just about the level of the pons. Once the signals reach the cochlear nucleus, then they split up and move contralaterally and ipsilaterally to an area called the olivary nucleus that we'll see is important for sound localization, and on up to cortex via the inferior colliculus first, or that part of the tectum, that inferior part of the tectum. And from the tectum of the midbrain up to the medial geniculate body of the thalamus, and from there up to primary auditory cortex on that dorsal surface of the temporal lobe. Now, I mentioned the olivary nucleus as one of the stops along the way, and remember that it gets information from both of the ears. This brings us to the idea of some of the mechanisms that are used to locate sounds in the world, and there are a few different approaches the brain seems to take. Particularly for higher frequencies, the brain seems to use information about the shadow that's created as sound passes through the skull. This is sometimes called a sound shadow, and it refers essentially to intensity differences. So, for example, if you were to put a speaker on the right side of someone's head, the sound would be slightly louder in the right ear than it would be to the left ear. There would essentially be a kind of a shadow created by the skull. So this information is thought to be used in some part to locate the sound on the right side. Other kinds of information that are thought to be used are time differences and phase differences. So for time of arrival, that's very much like the sound shadow. If you play a sound from the right side, it impacts the right ear just moments before it will impact the left ear. And this time difference is something that may allow us to locate objects, particularly when they're off in the periphery. The time difference doesn't really work when things are directly in front of or behind you, but at any movement laterally of these sounds, you'll see that the distance to the ears is going to be slightly different, and that's going to cause a slight time difference. The time difference and phase difference are somewhat related. The phase refers to the point of the oscillation in a sinusoid. So if you've got a compression that's moving along at this frequency, you can see that its phase is constantly changing as it moves through these cycles of the compression. What you'll notice, and it's thought that this might be more important for low frequencies, that for a nice big low frequency input, that the phase that it's in when it reaches in this case the right ear, is slightly different from the phase when it reaches the left ear. This phase difference can be used then to calculate the location of this input. The time of arrival method is somewhat similar in that it's simply using information about when action potentials are generated or communicated from the right and left cochlear nucleus as those signals come in from the cochlea. And it turns out that there are arrangements or networks of neurons in the medial superior olive, or olivary nucleus, I referred to it earlier, that respond to both inputs from the right and left ears. What I hope you will see from this is that when inputs are, for example, arriving at exactly the same time, so if there's a speaker right in front of you and your left and right ear both get that input at the exact same time, they will generate action potentials at the same time, and thus those signals will meet at this secondary neuron B. So they will generate action potentials, and their output or their excitatory postsynaptic potentials will both impact B at the same time. So B will become the most stimulated neuron. However, if the signal is slightly off-center, moved to the left or to the right, then we can get activations of neurons A and C. So if the sound arrives at the left cochlear nucleus first, then slightly later at the right cochlear nucleus, the signal from the left cochlear nucleus will have had more time to pass, and so it's likely that neuron C, for example, would become most active because it would be stimulated at the same time by the right, which was stimulated a little bit later because of the direction of the sound. So sounds coming from the left hits the left cochlear nucleus, the action potential starts. Moments later, the sound impacts the right ear, that action potential starts, and the two would meet here at neuron C. Likewise, for neuron A, you have the same kind of thing. And what you can take from these is that if you were to combine the relative activations of just these three neurons, you could infer the location of the sound from a whole range of locations. And it's thought that these small networks in the superior olive are important, again, for this rapid determination or estimation of localization of sound. So we'll skip through the rest of the thalamus and the inferior colliculus, which are, and I should mention that the inferior colliculus is another area that's thought to be important for sound localization and maybe generating reflexive responses that are related to the location of the sound. But once the information moves past the inferior colliculus and medial geniculate of the thalamus, then it moves on to this primary auditory cortex, which sits on that dorsal surface of the temporal lobe. And what's interesting about the auditory cortex is that it maintains what's called a tonotopic organization. So it responds in the same way that the cochlea responds to different frequencies at different locations, responds to different frequencies along its anterior-posterior dimension. So at the most posterior part of the primary auditory cortex, we have responses to the base of the cochlea, or higher frequencies, and then moving down anterior, we find that the primary auditory cortex responds more to the apex of the cochlea, or lower frequencies. In other words, again, like the cochlea, if you were to go in here with a microstimulator, you could essentially play the auditory cortex like a piano. Now, these neurons on primary auditory cortex respond to mainly pure tones, that is, individual frequencies. That's, again, this tonotopic relationship. As we move out from primary auditory cortex, the organization is very much like vision in that it's hierarchical. So we have simple responses to simple frequencies at the primary auditory cortex. Just like with the primary visual cortex, we had responses to very simple shapes. And then as we move out to the belt and parabelt regions, which are sometimes called the secondary auditory cortex, and then on into inferior temporal cortex and parietal cortex, what we find is that these regions respond to more and more complex kinds of auditory inputs. So the secondary auditory cortex, rather than responding to individual tones, tends to respond to maybe a range of frequencies or even sequences of or changes in frequency. So think of something like birdsong. So there are cells in our secondary auditory cortex, that respond to, for example, increases in frequency, so a change from low to high frequency. There are other cells that might like high to low frequencies. So these more complex auditory units tend to be coded next. And then we move on in this hierarchy into areas like in the visual system. So it turns out that the auditory system holds a what and where pathway or division that's very much like the visual system, and in fact overlaps quite a bit with the visual system. So in the auditory domain, this is sometimes referred to as the anterior and posterior pathway. The anterior pathway would be one that performed auditory processing on the inferior temporal lobe. The posterior pathway is one like the where pathway in vision that tends to process information about where sounds are coming from. Now, the way that this has been understood, if we look at single cell recordings, is that you could imagine individual cells down in the inferior temporal cortex that are processing the identity of sounds. These cells might respond selectively to a particular monkey call, for example. So in these cases, of course, individual cell recordings, these are usually done with primates. So a particular monkey call might cause to activate an individual cell in inferior temporal cortex. And it wouldn't matter where that call came from in space. There are cells then in the parietal cortex, or in this posterior pathway, that respond to the location of sound irrespective of what kind of sound it is. So it could be a cell that responds to any number of different monkey calls, but that cell prefers only those calls that come from a particular direction. So in this way, we see again this divide and conquer strategy, that the brain seems to take basic auditory information, break it down to simple units, and then piece it back together into its what and where kind of information. One of the last things about the auditory system that I'll mention is that it's again important. In these lectures, we often talk about these sensory systems as though they're somehow completely independent of one another, but it's the case that our sensory systems are all interleaved, and I hope you got a little bit of a sense of that when I mentioned that the what and where pathways for audition are overlapping quite a bit with those for vision. And that's because our sensory world is one that is entirely integrated. We don't have perceptions as we interact with people throughout the day where we disentangle the auditory and visual information. So you don't have, at least perceptually, right, this sense that, okay, well, I recognize that as my mom, but I'm going to wait till I hear her voice. That the voice recognition and perceptual or visual recognition, those things all are going hand in hand. You can seamlessly have a conversation with someone without second guessing the fact that the sound that's coming out of the speech that's coming out of their mouth is actually being generated by them, right? You have this visual perception and it's integrated with your auditory perception. What's interesting is you can see how the brain is wired for this when you tweak with the signals that are input to the nervous system. And this video from the BBC is one that illustrates that principle pretty nicely.
At any one moment, we are being bombarded by sensory information. Our brains do a remarkable job of making sense of it all. It seems easy enough to separate the sounds we hear from the sights we see. But there is one illusion that reveals this isn't always the case.
Ba, ba, ba.
Have a look at this.
Ba.
What do you hear?
Ba, ba, ba. Ba, ba, ba.
But look what happens when we change the picture.
Ba, ba, ba. Ba, ba, ba. Ba.
And yet, the sound hasn't changed. In every clip, you are only ever hearing ba with a B.
Ba, ba. Ba.
It's an illusion known as the McGurk effect.
Ba.
Take another look.
ba,
Concentrate first on the right of the screen.
ba,
Now to the left of the screen.
ba,
The illusion occurs because what you are seeing clashes with what you are hearing.
ba. In the illusion, what we see overrides what we hear. So the mouth movements we see as we look at a face can actually influence what we believe we're hearing. If we close our eyes, we actually hear the sound as it is. If we open our eyes, we actually see how the mouth movements can influence what we're hearing. Ba, ba, ba.
It's a bizarre effect. Remember, the only sound you're hearing is ba with a B.
Ba, 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 ba.
What's remarkable about this illusion is even knowing how it's done doesn't seem to make a difference.
Ba. The effect works no matter how much you know about the effect. I've been studying the McGurk effect for 25 years now, and I've been the face in the stimuli. I've seen stimuli thousands of times, but the effect still works on me. I can't help it. The speech brain just takes in that information and doesn't care about what outside knowledge you bring to bear. Ba, 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 ba.
The McGurk effect shows us that what we hear may not always be the truth.
Ba,
But it also helps us to understand what happens when our senses conflict.
ba, ba, ba. When the brain has the conflicting information, it tries to make sense of that conflict. And depending on what type of modality is providing more, salient information, that information might override or at least combine with the other information.
So we can't always trust what we hear, because sometimes our sense of vision takes over, enabling us to maintain a coherent view of the world.